Hi, welcome to Gut Questions. I'm Parker, friend and right-hand man to Dr. William DiPaolo. Will is an immunologist, and he's been studying the microbiome for about 10 years. Specifically, Will studies the bacteria that reside in the gut microbiome. He's up at the University of Washington in Seattle, where he is the director of CMIST, the Center for Microbiome Sciences and Therapeutics, and he runs his own lab. In this third installment of the PUFA episode, polyunsaturated fatty acids, Will explains the changes in the growth and virulence of Yersinia enterocolitica at room and body temperatures, and then at those temperatures in the presence of omega-6. In this project, they used an omega-6 called arachidonic acid. And what they saw is that the pathogenesis of Yersinia is changed such that it's a more virulent bug in the presence of that omega-6, the arachidonic acid. So again, this is an animal study, a mouse study, and the lab wanted to understand if the infection rate or the severity is increased when in the presence of the omega-6 at the room and body temperatures. So really pay attention to the whole bit in the beginning about the temperatures. That's key to this project. And with that, I give you my friend and boss, Dr. Will DiPaolo.
I mean, we found that, we were a little bit surprised because definitely the omega-6 had an impact on the growth. So that it actually, at the growth at room temperature and also the growth at 37. So it caused growth even when the bacteria is supposed to not be growing. So Yersinia mostly grows at room temperature, but we saw that with the addition of omega-6, we got growth even at
room temperature.
Body temperature.
Or body temperature.
Right. So we got omega-6 allowed it to keep growing, even though it was supposed to be turning on its virulence. And then at room temperature, when it's supposed to keep that needle, it hasn't made it yet. Well, we saw actually evidence that there was probably production of the needle and all of those virulence factors at room temperature. So even though like they hadn't got the temperature shift yet, they were still acting like they had. So that means that room temperature, these guys were actually more virulent.
Becoming more virulent because you've given them some omega-6.
And that somehow
they're like, we like that, and here's what we can do now.
Right. And so we don't know exactly what.
You're looking down at your dog.
No. I was I just found a bump on my knee, and I was thinking, what is that?
Because that's why I had been looking down and like.
My dog's upstairs.
Yeah. That was smart.
So basically, we found that at room temperature, they're more virulent, and then we decided to look to see. And how we did that was, again, there's a lot of technical stuff, and I can do a technical thing later. Like we can always do an add-on to this where I like talk about the techniques we use maybe for people who care.
Yeah.
Dive a little bit more into like what assays that we did to detect that. But basically we saw that arachidonic acid induces more of a virulent state in Yersinia. And that's at that room temperature. And so the next thing we did was, we saw that there was an increase in virulence. So then we wanted to see if we put the Yersinia onto human cells, will they be more infected with Yersinia? So Yersinia, when you ingest it in contaminated food, it will get to your epithelial cells and it will adhere to them. And then that adherence helps it translocate into your tissues. So it like goes from the inside of your intestines, the lumen, it'll move into your tissues through adherence. And so we wanted to just see, there's an assay that you do where you look to see if you can make cells. So you have a single layer of cells and you add your Yersinia on top of it. And then you basically, after a given amount of time, you wash it, and then whatever stuck is adhered. So then you can you can count the bacteria that remain in the plate that are adherent. The nice little trick that you can do with this sort of assay is you can do the same thing, but then you add a little bit of antibiotics to the wash, and it basically kills all the extracellular Yersinia And I told you earlier that Yersinia doesn't typically have an intracellular lifestyle, so it usually stays on the outside. But in some cases, Yersinia enterocolitica can live inside of a cell, but nobody understands why or what that helps do. And so we decided to just look to see if the omega-6 forced the Yersinia inside the cells just as a control. And so what we did find was that there was a significant amount of adherence that was increased with omega-6. And we actually found a very significant increase in the amount of bacteria that was inside the cells. So we actually are creating a more virulent bug, and we're changing its pathogenesis with the omega-6, forcing it into an intracellular lifestyle. And so what that means, we don't know. So we decided to then take that bug that we treated with arachidonic acid, and we treated it, we washed it away so that we just took the bug itself. And we did an infection study where we gave it to mice to look to see if the mice would, comparing mice infected with regular Yersinia grown at 37, and then in given to the animals orally, like you would ingest it, and then, or the Yersinia enterocolitica at room temperature induced with the omega-6.
Omega-6.
And given to the mice. And we looked to see what would happen. And we found that the omega-6 treated Yersinia caused a much more severe infection. It was pretty rampant by day. So a normal Yersinia infection in rodents would basically, they can live, depending upon the dose, but like 6 days, 7 days, like until you start to see signs of infection. With the Yersinia enterocolitica treated with the omega-6, they were sort of showing signs of infection at 2 days.
2 days.
2 days, yeah, 2 days instead of 6. And then we had to end the experiment on day 3 because of how the mice were progressing. So
I'm curious to now go to look at figures, because I feel like the figures will make so much more sense to me.
Right. So basically, and then we did find, as a little bit of an aside, we did find that omega-3 could inhibit omega-6 in some of these cases. So it inhibited the growth of the bacteria if you put omega-6 with it. So omega-3 would inhibit the omega-6 induced growth we saw. And it also—
And you would do this both at room temp and at body?
We did everything at room temp and at 37, but really we saw the greatest effects at room temperature.
Okay.
Yeah, at room temperature. And then, we did some multi-top experiments looking at protein things, which I think is out of the context of this, but just basically this omega-6, treating these Yersinia with omega-6 changed the metabolic fingerprint of the Yersinia enterocolitica, and it's thus leading to an increase in virulence factors and an intracellular lifestyle that could—
because things that they normally do, they're either doing or not doing anymore, which is allowing them to behave
differently
in a different way.
Yeah, you're giving them a source, like something that they may not need, or they need only a small amount of, and so then you're forcing them into a metabolic program that's different, and part of that metabolic program could be this intracellular lifestyle. And so right now, we didn't get around to doing the RNA transcriptomics, so we wanted to basically do a screen to look for all of the sort of pathways that were induced, and so we're still working on that, but we're submitting the paper without that, and hopefully the reviewers don't listen to this podcast and then say, oh wait, that's a good idea, and ask us to do it, because we haven't yet. So I'm still doing that process, but this was all really just to prove a point that, I think in science, we don't tend to look at how these sort of molecules affect bacteria specifically. I'm not saying everybody does that, but I think as far as like dietary factors go, and like these sort of dietary nutritional components and things like that, the measurement has always been a readout of host of human response.
Right. Not what is changing with the bacteria.
Right.
Like you give that bacteria something, it'll use it and produce something different. Like their metabolites, is that the right word? Or their byproducts or what have you will be totally different.
Mm-hmm.
And then those are now in your system.
Right.
And or they might not produce metabolites that they should be producing. Is that accurate too? Or not necessarily accurate, but a thought?
Right. So we don't know how the being forced down this metabolic pathway, how it's going to trigger or inhibit the normal metabolic pathways that would potentially be activated in the absence of that. So that, I think it's a good question. I'm very excited to do that experiment, looking at how pathway experiments. We have everything to do it. I think I just have to talk to somebody in the lab into doing it. But
like Marie Pierre, would she do it? I want to know now. I think I like this project so much more. I was a little overwhelmed at times.
Well, yeah, so it makes, I mean, it makes sense, and it's like, it's a nice project. And I think
it's very like simple but elegant, you know, yeah, like it's just so. It's not a hard concept, but the way that you have pieced it together, you and Denise, it's really nice.
Yeah, and I think, right, and I think that it just gives a nice little point. It's a very small little story from the lab. It's like, you know, it's like 4-figure paper we're just trying to put out there.
Yeah.
Just, and like really the idea was this was actually a branch off of a bigger project that was looking at an animal model of infection, where we were treating the mice with these different diets that actually had these different components, and then giving them an infection and then looking at it. And that's a mixed bag, because you have that's exactly what I was just talking about. You're going to have a lot of readouts that are host cell, and to discriminate what's actually happening from the bacterial side and what's happening from the host cell side is much more complicated, because you have both working together. So like this was a side, like we just said, well, let's just take the pathogen and just see what happens there.
Yeah, break it down.
So yeah, so then the next step is to go back and to start looking at the infection process in these guys, and looking in how the host responds to these bacteria in the context of an omega-6 heavy diet, a Western diet.
Well, yeah, okay. So I was completely lost on this project, I will admit. And I will also admit that no, I didn't speak up or try and understand it, because honestly, I do other things for Will. But I would just get bored, to be frank. When scientific talk starts to happen, I just kind of tune out with all the acronyms and the jargon. And even though Denise is very clear, she speaks beautifully, her slides are beautiful, I tune out. But being able to have this one-on-one with Will and interject with my questions at any moment, I've found that while I understood the project to some extent and could appreciate that it was really just good, solid science, I now have a much better understanding, and I see the importance of this project, which is actually what Will and I will talk about in the last installment of this episode. Will talk about the relevance of this project to our everyday lives. So definitely check that out. And if you've got questions for Will, please email me at aparker@medicine.washington.edu, or leave a comment in the comments section below, or visit our website at yourgutquestions.com and send me a note that way via the contact page. See ya.